So hi guys, let's discuss a question today. So this question is a very interesting question because it was one of the NEET-PG recall question and this was the question. So let's look at the question here. So basically the question was which technique is used to measure CD markers. Okay, so when we talk about CD what is CD? Cluster of differentiation. So there are some antigens which are present on the cell surface. Okay, so whenever you want to identify them, you have to use a fluorescently tagged antibody which will go and bind with them and it will give fluorescence. So if they are present, you will see fluorescence. If they are absent, you will not see fluorescence. So let's look at this case here. So you can see the fluorescence is beautifully coming in CD10, CD19, CD34. So this is a typical case of B acute lymphoblastic leukemia. You understand? So whenever we have to study the cluster of differentiation, the cluster of markers that are present on a cell. So it not only identifies one cell, one antigen, it can identify the many antigens together in one go. So whenever you have to identify what is the immunophenotype in the flowing cells, so that is why flow means flowing cells, cyto means cell, metry means measurement. So whenever you want to analyze what is the total antigenic composition of a cell, we use a technique called as flowcytometry, and this is also called as immunophenotyping. Okay, so that is one thing which you have to be clear. So that is your flowcytometry. Now, what was the other options? The other options were again very simple, but you have to understand. So the next option was cytogenetics. So cytogenetics name itself says cytogenetics, cytochromosome analysis. So whenever you want to analyze any abnormality or you want to identify the chromosome number and anything in the chromosome structure, okay, like your translocations or if you want to identify that what is there an aneuploidy, any number differences, trisomy, triploidy, then you use cytogenetics. And most common stain that we use in cytogenetics is GIMSA. That is why it is also called as G banding. So that is for chromosome number and chromosome alterations. Okay, so that is cytogenetics. Coming to the third technique. Third technique was immunohistochemistry. So name itself says immunohistochemistry. Histo means histology. So whenever you want to identify the antigens, okay, the presence of the antigens in the tissue, okay, then you use immunohistochemistry. And immunohistochemistry has a simple rule. Okay, what is the simple rule? That if that protein is present, so basically it is for identification of protein or antigens on the cells. Okay, on the cells, remember that. So if that protein is present, it will give a positive brown color. If it is absent, it will not give any color. So here is a all or none phenomena. So if you're seeing that brown color, it's positive. If you're not seeing any brown color, it is negative. So that is immunohistochemistry, which can show you variable patterns. So it can be either in the membrane, like in HER2/neu, or it can be in the nucleus brown color, that can be ER, PR, okay, that is nuclear marker, or Ki67, which is a marker of mitotic activity, it comes in the nucleus, or in the cytoplasm, like intermediate filaments, all the cytokeratin, vimentin comes in the cytoplasm. So the rule is all or none. If you see brown color, it is immunohistochemistry positive. Now, coming to the fourth technique, what is that? ELISA. Okay, so what is ELISA? ELISA has variable generations, and whenever we want to identify the soluble antigens or antibodies, okay, soluble, then we use immunohistochemistry. So soluble in the plasma, okay, or you want to take out the blood and you want to analyze what is the soluble antibodies which are present in it, like let's say serum iron you want to measure, you do it with ELISA. Serum TIBC you want to analyze, you do it with ELISA. Understood? So whenever you have to analyze anything in the serum, okay, soluble proteins you want to identify, either antigens or antibodies, that is done by ELISA. So here is how the ELISA comes. Okay, depending, can have various color depending on the chromosome that you're using. Okay, so that are 4 common techniques that you have to be very clear. Okay, so whenever you want to identify all the antigen composition of a cell, of a flowing cell especially, so that is flow cytometry. If you want to see what is the protein which is present on the cells, on the tissue, on the histology, that is immunohistochemistry. If you want to identify the soluble antigens or antibodies in the serum, okay, in the serum, like serum iron, serum TIBC, that is your ELISA. And if you want to analyze the anything in with chromosome number, structure, that is cytogenetics. So each technique has its own advantage and disadvantage, but first thing first, you have to understand the basic techniques first. So now can you identify and answer this question? So if I'm giving you this question, I'm sure all of you know the answer now. So this is cytogenetics. Okay, so that's a basic thing that an undergraduate should know, and you should be very clear with this. Right guys, bye.